Hi guys.
Good morning, Mr.
Good afternoon. حابين في الصيام. صايم يا احمد ولا فاطر؟
الحمد لله مستر صام.
فاجئتني. طبعا مروان فاطر. اوكي. بسم الله. Today we are going to complete the new topic, which is biotechnology and genetic engineering. In last session, we explained the difference between the term biotechnology and the term genetic engineering. Biotechnology means using of living organisms or their hormones or their enzymes for producing some products or making or develop some technological applications. But genetic engineering, it is related to the genes of living organisms, like changing, inserting, removing genes from species, or inserting gene from species to another one, but this inserting of genes just for improvement and development some traits or production of some products, and we can consider that genetic engineering similar to selective breeding, not identical to each other, but is slightly similar to selective breeding. And then we explain the examples about biotechnology, like using of yeast fungus in different productions, like producing biofuel, making bread, making alcohol or brewing, and using of enzymes from some living organisms for extraction of juice from fruits, like pectinase enzyme, or making washing powders, detergents for removing some stains from clothes, and production of antibiotics by using some living organisms like fungus. And the most common example about this antibiotic, like production of penicillin. And the last one, the genetic engineering, for example, the production of insulin for the diabetes disease. We explained that the uses of yeast fungus for different production, making bread, production of brewing or wine and alcohol, and the biofuel depends on the type of sugar. For example, Uh, for making bread, start of
Glucose.
yes, start of aerobic or anaerobic respiration uh, on the glucose sugar. But in case of making brewings, wine and alcohol depends on, for example, for making alcohol or ethanol, depend on the maltose sugar from the barley seeds for making alcohol for beer. And for making wine depends on the sugar from the grapes fruits. But for the production of biofuel depends on the type of sugar in maize plants, type of sugar in maize plants. And then we mentioned the difference or the advantages and disadvantages of biofuel, ethanol-like biofuel, or the fossils biofuel. And then we explain the using of enzymes for production of washing powders, detergents, for removing stains from clothes. But firstly, we said that the living organisms from which will extract the enzymes. Firstly, these living organisms or microorganisms put inside fermenters. During or what happens inside these fermenters? These fermenters provide the microorganisms with oxygen, with nutrients, with a suitable pH, with a suitable temperature to help these micro living organisms to perform its own life functions normally, so start to secrete the enzymes which will used for making this washing powder. The most two enzymes used or enter the structure of washing powders, lipase and protease. Protease, this one for breaking down proteins, and lipase for breaking down lipids. And both of them used for removing the stains, which includes proteins and fats or lipids. And then we mentioned the disadvantages and advantages of uses of washing powder. And then we explain the extraction of juice from fruits. This extraction depends on enzyme called pectinase enzyme. Pectinase enzyme. This pectinase enzyme have the ability to break down the pectin substance, which we can consider that enter the structure of the cell wall of the fruits. So this pectinase enzyme have the ability to break down this pectin enzyme, so it break down and release the cell wall of the fruits, so it make the extraction of juice more easily. So pectinase used to extract juice from fruits, so it make it more easier to squeeze that juice from the fruits. The last point in this topic, the genetic engineering. Genetic engineering depend on the genes. Yes.
You said what does pectinase do to make it easier?
pectinase used to break down the pectin substance. This part, pectinase, okay. Break down the pectin substance. Pectin substance, we can consider it similar to intermolecular forces inside the molecules, or we can say similar to the structure of cell wall, or simply we can say that pectin substance hold or collects the cells of the fruits together. By breaking down this substance, so the forces between, or the binds between the fruit cells broken down, so it makes the extraction of juice or squeezing of juice from these cells more easily. Clear?
مستوعب.
What?
هي موجودة في أنهي أورجان البكتينيز دي؟
Again, please.
البكتينيز دي في أنهي أورجان؟
Pectinase enzyme not in human.
أوكي.
We digest the fruits.
أوكي.
Pectinase enzyme substance extracts from some insects and some reptiles. They depend on fruits, and this pectinase enzyme break down the pectin substance, which between cells or hold the plant cells together, so it makes the extraction of juice outside the fruits more easy. Is it clear? Hala, you got it? Hala? What?
Yes.
Yes, okay. So what is the pectin substance? You can explain it?
Substance.
Mm.
Substance that holds cells together. It is something like cell wall, or it has the structure of cell wall, or it's similar to forces between the cells.
Yes. Forces between molecules, not cells. Forces between molecules, not cells, okay? You hear me, Hala?
Yes.
So forces similar to the forces between the molecules, not between cells.
Okay.
Between cells, substances hold the cells together, not force together. Okay, the last point, genetic engineering. Genetic engineering means simply we can say that we can take gene from living organisms and put it inside other living organisms for improvement or for development or production of technological application. But we can perform genetic engineering in the same living organisms without adding or inserting any genes from other one. We can make it by removing by changing the sequence of the genes. We know that all living organisms have sequence of genes, its own sequence of genes. Let me ask you, the sequence of genes which is responsible for the appearing of different traits. Similar in living organisms, we can say that Omar and Hanin, they are identical or non-identical twins. Identical or non-identical?
Can you repeat the question, please?
Okay. Omar and Hanin, identical twins or non-identical?
Non-identical.
Non-identical.
Unidentical.
Again, identical or not?
مش not.
Not.
Not identical, right?
Mm.
Clear. So they have the same traits or different traits?
Different.
Different. So they have same genes or different genes?
Same.
Different.
Different.
Different because they're non-identical.
Okay. So in case they are identical, they will have the same sequence of genes or a different sequence of
I
genes?
think they will have the same sequence of genes
Why?
لا.
because they are identical, have the same genes.
Because they have the same traits, and the same traits appears by specific genes.
Same genes. Yes.
Yes. So in case of identical twins, they have similar sequence of genes. In case of non-identical twins, they have different sequence of genes. In case of changing the sequence of genes, this sequence still producing or appears the same trait or will be appears different traits. By changing the sequence of genes, the traits still as it or will it change? يا دكتور حلا.
Change.
Will it change? Right?
Yes.
So, يا دكتور حنين, if we change your genes, for example, the genes responsible for eye color, we can say that you have genes responsible for the dark brown eye color. By changing the sequence of these genes, you will still have dark brown eye color, or the eye color will change as a result of the changing of these genes.
Change.
Change. In case of a changing the sequence of genes, changing the traits, so we can improve some traits in living organisms. So in this case, we can consider that this is changing of the genes, genetic engineering or not.
Yes.
Yes.
Yes. Yes. So genetic engineering is not just inserting gene from a species to another. We can make genetic engineering in same living organisms without adding any external genes. By what? By
Can you please repeat
removing,
this?
again, I hear you.
Sir, I'm saying, can you please repeat this part? This
Okay.
sentence.
Okay, I just need from you to know that genetic engineering, it is not just depending on the inserting gene from one to another. Take gene from Omar and put it inside Hanin, for example. Genetic engineering depends on other methods. By what? By changing the sequence of the genes in same living organism, or removing genes from living organisms. For example, if we know that a specific gene responsible for some disease or a disease, so we know that this gene affects the appearance of this disease. By removing this gene from the living organisms, the disease will occur or not? Move gene, we can change sequence of genes for improvement or development the living organisms. You got it?
Yes.
So genetic engineering, just inserting gene from one species to another?
No, inserting, removing, or changing sequence of genes.
Excellent. بس ده اللي انا عاوزه. All of you, you got it? It's clear? دكتور عبد الله.
يس يا مستر الحمد لله.
يس الحمد لله. انا ما سالتكش انت عامل ايه، بس تمام، انا طمنت عليك. انت فهمت؟
شوية يعني.
شويه. ايه اللي انت مش فاهمه؟
لا وانا خلاص يعني فاهم الحمد لله. لا هو
طب انت فاهم الجزئيه بتاعت الجيناتيك انجينيرنج؟ طب ممكن تقول لي كده سمري بسيط كده عن الجيناتيك انجينيرنج؟ فكرته ايه؟ فايدته ايه
فكرته
كده؟
اللي احنا يعني بنحط جينز في living organism معين.
جميل.
و
طب انا هعمل inserting gene from one species to another for what؟ يعني انا هجيب جين من حد احطه في حد تاني ليه؟
عشان نعمل change للcharacteristics.
change للكاركترستكس، يعني change للكاركترستكس كده وخلاص؟ ولا انا مثلا عايز اطور حاجه، احسن حاجه، عايز اعمل حاجه جديده؟ انت شايف ايه؟
لا اطور حاجة.
يبقى genetic engineering aims to improve or develop or protect the living organisms against some disease. يعني مثلا انا عرفت ان في جين معين لما بيبقى موجود بيعمل مرض. طب انا بقى خلاص عرفت ان الجين ده بيسبب الديسيز ده، اسيبه ولا اغير فيه؟ انت شايف ايه؟ ولما اغير فيه هتجنب المرض، صح؟
صح.
يبقى انا طورت كده الليفنج اورجانيزم ده وحميته من الديسيز ولا ما حميتوش؟
حميته.
وبالتالي هيبقى عنده كاركترستكس افضل ولا الاول افضل؟
ده يبقى عنده افضل طالما انا حميته.
بالظبط كده، هو ده بقى الايمز بتاعت الجينيتك انجينيرنج. بس البارت اللي انا بقول فيه بس ان الجينيتك انجينيرنج inserting gene from species to another، يعني أنا مثلا production of insulin مثلا، ده الاكستراكشن بتاعه مثلا بيجي من الهيومن، عارفين ان الهيومن بيعمل سكريتنج للانسولين. مين عارف اني اورجان يا شباب بيعمل سكريتنج للانسولين؟
للايه؟
الانسولين.
Pancreas.
Who said it? Jana, right?
Yes.
Yes, pancreas. Pancreas secrete insulin to control the level of sugar inside the blood. So,
Mister,
yes,
could you please repeat the question?
which organ secreting insulin?
Okay.
Which organ?
Pancreas, I don't know.
Pancreas, right? Okay, and pay attention, please. Abdullah, you hear me, right?
Yes.
Okay. The diabetes disease, it's related to the level of the sugar inside the blood. So this disease require take insulin. الناس اللي عندها السكر تبقى محتاجة تاخد انسولين، صح؟ صح يا شباب؟
اه.
ليه؟ لأن ممكن يكون هي ما بتعملش سكريتنج ليه ب يعني enough quantity، أو إن هي البنكرياس بتاعها مش مور أكتف، ففي مشكلة فما بيعملش سكريتنج للإنسولين بنسبة كويسة، فتضطر إن هي تاخد اكسترنال انجكشن، تاخد حقن، صح كده؟ صح؟
يس.
فأنا عشان أعمل حقن انسولين دي يا عبد الله، تفتكر امسك واحد واحد بني ادم بني ادم اخد الانسولين اللي في جسمه عشان اعمل منه حقن؟ ده صح ولا غلط؟
لا غلط.
غلط.
غلط طبعا. فهضطر اعمل ايه بقى؟ تفتكروا هنعمل ايه؟
هناخد كمية كافية من واحد مثلا.
هنعمل اكستراكشن للسيكونس بتاع الجين اللي بيعمل الانسولين، يعني ده هرمونز، والهرمونز ميد اوف بروتين، والبروتين دي ليها جين معين، ليها الاستراكشر بتاعها. فانا لما اخد الجين بتاعتها او طريقه عملها بقى كده بالبلدي من الهيومن، واعمل لها انسرتنج في ليفنج اورجانيزمز زي البكتيريا، والبكتيريا سهل ان انا ازود عددها. لو انا مثلا جبت some bacteria and put it or put them في suitable medium, suitable temperature و suitable pH و more oxygen و more nutrients، فدي هتبقى مكان كويس ان هم عددهم يزيدوا ويكبروا، صح؟ صح يا شباب؟
هو
صح.
حضرتك بتقول ان انا هاخد الانسولين ده هاخده من حد وبعدين اروح حاطاه في بكتيريا، البكتيريا هتعمل له ايه؟ هتزوده ولا مش فاهمه؟
مش هتزوده، انا هاخد
اه.
الجين، انا ما بقولش هاخد الانسولين، انا هاخد الجين
اه.
اللي هو مسؤول عن تكوين الانسولين، اللي هو بالبلدي كده طريقه عمل الانسولين،
ايوه.
واخد الجين ده احطه في بكتيريا. كده بقى البكتيريا عندها الجين اللي بتعمل ايه؟ امم
بتعمل الانسولين.
جميل. والبكتيريا دي لما
ايوه ممكن.
احطها، اسمعيني، والبكتيريا دي لما احطها في مكان سيوتبل لللايف بتاعتها وتعمل ريبرودكشن وعددها يزيد، كده الجين ده فضل موجود في نفس البكتيريا اللي انا حطيته فيها الاول، ولا هيتنقل لجنريشنز ثانيه؟
هيتنقل.
هتنقل.
جميل، هيتنقل، برافو. ولما يتنقل هيبقى عندي بكتيريا كتير عندها انيجين اللي هو بيعمل الانسولين، فيبقى كل البكتيريا اللي انا خلاص بقى عندها الجين ده تقدر تعمل بروديوسنج للايه؟
انسولين.
للانسولين. صح. سهل بالنسبه لي اخد الانسولين من البكتيريا دي ولا اخد الانسولين من الانسان؟ مين الاسهل؟
من البكتيريا.
من البكتيريا. حتى لو هي ماتت مش هتفرق معايا في حاجه، صح ولا غلط؟
صح.
يبقى انا الجيناتيك انجينيرنج هنا ساعدتني ان انا اعمل انسولين او برودكشن اوف انسولين. ازاي؟ عملت ايه؟ خدت جين responsible for the structure of insulin. ما خدتش الانسولين نفسه يا شباب، ما خدتش الهرمونز نفسه، انا خدت الايه؟ الجين. يعني ايه خدت الجين؟ يعني الجين اللي فيه طريقه عمل الايه؟ الانسولين. صح كده؟ It's clear?
Yes.
And then inserting this gene inside other microorganisms like bacteria. So in this time, this bacteria have the gene which responsible for the structure of insulin to control the blood, the level of sugar inside the blood. And then this bacteria, by making a suitable medium to their life functions and suitable pH, suitable temperature, suitable oxygen, suitable nutrients. So this bacteria starts to reproduce. And then this gene transferred from generation to another. So in this time, we have a lot of bacteria producing insulin, and then by extract this insulin from this bacteria or their bodies, we have the ability to make injection insulin to treatment the diabetes disease. So this idea depends on genetic engineering. You got it?
مستر
Yeah.
Yes.
هو طول ما انا شغاله بجينز يبقى ده
هو الكلام اتقطع فما سمعتش.
طول ما انا شغاله بجينز يبقى ده جينيتك انجينير.
طبعا.
غير كده يبقى
غير كده ان انا هستخدم ايه مثلا؟
البايوتكنولوجي انزيمز مثلا.
يس.
انزيمز وايه تاني؟
يعني لو خدت ليفنج اورجانيزم نفسه عملت بيه حاجه يبقى بايوتكنولوجي. يعني مثلا خدت اليست عملت منها بيكنج بريد يبقى بايوتكنولوجي. انا خدت الايه؟ الاورجانيزم نفسه او سيستم منه او برودكشن منه، زي مثلا الانزيمز. لو انا خدت منه انزيم، ما خدتش جينز، خدت انزيم، يبقى بايوتكنولوجي. لما ابتدي استخدم الجينز بقى يا حلا يبقى انا كده مش بايوتكنولوجي، انا جيناتيك انجينيرنج. You got it? Is it clear? It's clear?
يس.
Okay. So genetic engineering depend on the genes in living organisms' bodies, but it's not just inserting the gene from species to another, we can perform genetic engineering by adding or changing or removing genes from species. And we can use the genetic engineering to improve some plants against herbicides. For example, pay attention, please. Soya bean, the soya bean. This plant modified by genetic engineering to resist herbicides.